Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Okay, today in this video lecture we are going to talk about another topic which we call that post-transcriptional modification system of RNA. We also call that co-transcriptional modification of RNA, and we are also called that RNA processing or editing. Now, what are this process means? It is basically a biological process which are mostly occur inside the nucleus or which are mostly occur inside the eukaryotic cell. And due to this process, the RNA which is synthesized, the RNA are get matured. The RNA which are synthesized during transcription are get matured. There is some changes occur in the RNA. The 5' capping, which we called it, the changes may be 5' capping, the changes may be polyadenylation, poly A tail, the changes may be splicing. So whenever these kinds of changes or modification occur to the RNA, then this RNA get matured, and when this RNA leave the nucleus and reach to the cytoplasm and ready for the translation, or when this RNA is ready to produce protein, then this process is called as post-transcriptional modification, or we call it co-transcriptional modification, or we also call it RNA processing or editing. So remember, post-transcriptional modification or RNA editing, it mainly involved in three main stages. Okay, so there are three main stages due to which the RNA is modified. So these three modification are, let me write here. The first we call that 5' capping. Let me write 5' capping. Second we call that polyadenylation. We call that polyadenylation, means 3' polyadenylation of 3'. And the third we call that splicing. This is the first video lecture of this post-transcriptional modification of RNA. And in this first video lecture, we are going to talk about the overview. So let me write. Or the summary, concept, or the basics of this post-transcriptional modification. So in this video lecture, we are going to talk about these three basic modification, these three changes in the RNA molecule. So how these three changes will occur, we are going to talk about each three things, each three process, 5' capping, polyadenylation, and splicing. And inshallah, in the next video lecture, then we are going to talk about each of the three things in step-by-step video lecture in our detail. Okay, so first of all, we need to understand the concept behind that, the basics behind that what actually 5' capping means, and what are the means of 3' polyadenylation and splicing. Okay, so once we get an idea about the concept, and once we get idea about the big picture of this post-transcriptional modification, these three things, and then inshallah in the next video lecture, when we are going to explain the detail about that, then it will be very easy for you. So what actually happened, again, if we explain it, so let me draw again here. So first of all, let's explain that the 5' capping. What actually happened, again, we have, the RNA. So as you can see. So let's suppose this is the RNA. So let's suppose this is a DNA molecule. DNA, double-stranded DNA. And during transcription, this is not actually the transcription part. This is not the DNA transcription part. But we can say that somehow this is the co-transcriptional event, we also call it. What actually happened during transcription? This is the RNA polymerase, let's suppose. So during elongation, what happened? The RNA strand are synthesized. So this red one can represent the RNA strand. If you don't know about the elongation of DNA transcription, so here I will recommend you that go back to my channel and watch the video about the transcription in eukaryotes. And you will check the three main stages: initiation, elongation, and termination. So you need to understand each stage. Once you get idea about DNA transcription, then this one video will be very easy. So what actually happened during elongation? The RNA strand are elongated. So this is the RNA strand having 5' to 3' end. This is the 3' end. And this is the DNA strand, so its one strand should have 5 to 3' end, while its other strand should have 3 to 5' end. So mainly the RNA polymerase 2 or RNA polymerase, actually we are going to talk about the RNA polymerase 2 mediated transcription. So what actually happened, the RNA polymerase 2 will use the 3 to 5' strand, having those strand having 3 to 5' end. So they will use those strand of the DNA and then it will produce the RNA strand. Now after the addition of 60 to 70 nucleotide, RNA nucleotide, after the addition of 60 to 70 RNA nucleotide, what happened? This at this 5' area of the RNA, there will be 3 main things or 4 main things will occur. So the first we call that guanine, okay, and the second we call that methyl group, and we also have some kinds of enzymes, proteins, which we call it triphosphatase. So these kinds of enzymes are involved and they will, each things are combined together. So first of all, the triphosphatase enzyme will bind here because there is a phosphate group. So they will cleave the phosphate, and after the guanine will add it here, and after that the methyl group will add it here. So when these 3 things are working together, then what actually happen, at the 5' end there will be a capping occur. So the 5' area will be modified, as you can see. So when the 5' area is modified, we call that 5' capping, or we also call it capping. So how exactly it will be going on, what is guanine, we will see inshallah in the next video. We will see the guanine structure, and in the guanine structure we will see the methyl group is attaching, and we will see that how the what kind of enzymes we need, triphosphatase usually we need in this case, and how it cleave the phosphate group, what actually going on, we will explain it in a detail. So this is actually the concept behind that 5' capping. Okay, now after that, once actually this 5' capping mainly occur after the 60 to 70 nucleotide addition. Why? Because 5' area is already free. Now once 5' area is free, so then capping will occur. After that, the 3' area should be modified. So it is not modified directly, it should be free from the DNA strand. So after the RNA polymerase synthesizing the 3' area, so it will reach to a kind of signal which we call a termination signal. We already explained the termination signal. So here I again recommend you that go back to my channel and watch the termination of DNA transcription, then you will get idea about that. Now what actually happened in termination of DNA transcription, we have termination sequences and termination signals. So the termination sequences are like that: A, U, A. These are the termination signal. And we have termination sequences like T, A, T, C, G, T. These are the termination sequences. Okay, so the termination signal are found on the RNA. These sequences, termination signal is nothing but is a kind of sequences, as you can see, which are found in the RNA strand. And termination sequences are the sequences, as you can see, which are found in the DNA strand. So when both are encountered by the RNA polymerase, then what actually happen? In the RNA polymerase, we already talked that there is the C terminal of RNA polymerase, which contain 2 kinds of the protein. One we called it CSTF and other we called it CPSF. Cleavage stimulatory termination factor, cleavage polyadenylation or polymerase specific factor, like that name. These are the 2 factors, or we can say termination factors, which are mainly found at the C terminal of this RNA polymerase. So it will be activated when it encounters the termination signal as well as termination sequences. Then it will get activated, and these 2 proteins, as you can see, will shift it to this one area of the RNA. So let's suppose this is the first protein, as you can see, which can bind on the termination sequences, and this is the CPSF, so it will bind here. And then we have another this is another term, we can say that termination. This is another cleavage factor. So this cleavage factor, which we call it CSTF, it will bound at that specific area having the CA region. So here there will be a CA sequences. So it will bind at the CA sequences, and we call this cleavage factor is called as CSTF. So when both are banded here, then it will recruit some other additional cleavage factors. So some other additional cleavage factor, a lot of additional cleavage factor will bind here, as you can see. So what are the function of each cleavage factor? We call it CSTF because cleavage stimulatory termination factor, these are those factors which can stimulate some other additional termination factors. Due to this way, it will make a cut after the CA sequences. So with this AA, there are some other nucleotide which we call it CA. So after the CA, this CSTF will make a cut. Now after the cutting, the RNA strand will be get freed. So this is the RNA strand. So now it is free, as you can see. So this area is already capped, or this area is already methylated capped. And this area, which we call it 3' area, it is already freed. So after that, the additional cleavage factor and CSTF will make a cut at that specific area. Although the cut is also made at that specific area as well as at the 3' end as well. So at somewhere, let's suppose at that specific area, a cut is made. Okay, due to this cutting, then what actually happen? Polymerase A. Let me write polymerase or polymerase enzyme. It will get activated, and once it activated, so what are the main function of this enzyme? It will add adenine nucleotide. So almost 200 adenine nucleotide are added at the 3' end. So this is the 3' end, and it is added by polymerase enzyme. And after that, poly binding peptide or like that kind of protein will bind to that specific area. That will scan the amount of the adenine. So when the amount of adenine reaches to 200, then this one PBP, which we call a PBP protein, will get released out. Okay, so this is called as 3' polyadenylation. So second process is 3' polyadenylation. Next, what actually happen? We have inside this, this is actually not the mature RNA. Inside this RNA, which is synthesized after the DNA transcription, inside this RNA, what actually we have? We have a specific kinds of region. So let me draw here. Let's suppose this blue region, as you can see. So we have a specific kinds of region inside the RNA, as you can see, we call that exon. And then we have some other specific region which we call that intron. Okay. Now what are the exon and intron? Let me a little bit tell you about that. Exon are the coding region of the RNA strand, which is present in the RNA strand, which are mainly responsible to give signal for the protein synthesis. Intron are the non-coding area of the RNA strand, which don't participate in the protein synthesis. So what happen? This intron should be removed. These intron portion will be spliced out, or splicing will occur. This, now what actually happen, let me draw here. Okay, now these intron are become spliced. Now this structure, as you can see, we call that spliceosome. These structure, as you can see, we call that spliceosomes, or spliceosome is also an organelles. Now the spliceosomes once formed, then it will be chopped out, it will be cleaved out, as you can see. So once all the intron portion are removed out, so it will be removed. So once each portion are released out, so only axon portion is remaining, and each axon portion are then connected to each other, as you can see. Now these blue are the axon portion. At 3' we have adenine, 200 adenine residues, and the 5' area we have a capping. So now this is the mature mRNA. After some time, this mature mRNA, all these processes will mainly occur inside the nucleus. And once mature mRNA are formed, so then it leaves the nucleus and transported to the cytoplasm or cytosol of the cell. And after that, it is ready for the protein synthesis. And after that, protein will be synthesized, or we can say translation will occur after that. Remember one thing, this is called splicing, okay? Now remember guys, this post-transcriptional modification will always occur inside the eukaryotic cell. It's not occur inside the prokaryotic cell. And why this post-transcriptional modification system is very important? Because what actually happened when we provided the antibiotic or the drugs, then what actually happened, this antibiotic drugs don't find any of the target area. We have some kinds of antibiotic which have the target area, which have the target area 5', which have the target area 3'. Okay, so these are the targeted area. So inside the bacterial cell, as the post-transcriptional modification doesn't occur inside the bacterial cell, so what actually happen, the antibiotic will degrade that specific area as well as it will degrade the 3' area. Now, as inside our own RNA, post-transcriptional modification will occur. So 3' area is also modified, means adenine will added, and 5' area is also modified, means capping will occur. Due to this way, and sometimes splicing will also occur. Due to this way, antibiotic don't have the ability, they didn't identify the 5' area as well as the 3' area. They don't have the ability to identify this area. That's why antibiotic doesn't degrade our own messenger RNA. So they will degrade the bacterial messenger RNA. So that's why this post-transcriptional modification inside the inside the eukaryotic cell is very important. So this is all about the post-transcriptional modification of the mRNA, or we call that RNA editing, RNA processing, or co-transcriptional modification system. Inshallah, in the next video lecture, we are going to explain the 5' capping in a detail. We are going to see the guanine structure, methyl group addition. We are going to talk about the protein or the enzymes, and we are going to see the mechanism of each 5' capping in a detail in our next video lecture. I hope you guys get idea about that, but if still you have any kind of confusion or question in your mind, you can write your question through a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and also subscribe the channel to get more interesting videos like that. And stay tuned, because in the next video lecture, we are going to talk about 5' capping. Thank you so much for watching.